A very good morning to all the delegates present today. Today, this is our first webinar of academic year 25-26. I welcome you all to Innovative Horizons in Pharmaceutical Sciences webinar by Gokhale Education Society's Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research. Myself, Dr. Shilpa Harak, is here to introduce you to the speakers to the session of today. Before we go forward, I would like to speak something about our society. The Gokhale Education Society was established on 19th February 1918 by Principal T.A. Kulkarni, who was a great social worker on the occasion of third death anniversary of his spiritual guru Namdar Gopal Krishna Gokhale, who was the spiritual guru of Mahatma Gandhi. The society has completed 107 years of meaningful existence and today has 140 units spread over three zones of Mumbai, Nasik, and Palghar, and have a students of 1.25 lakhs every year. Previously, it was led by late Principal S.B. Pandit Sir, President, a great visionary and eminent personality, and late Sir Dr. M.S. Gosavi, Secretary and Director General, who rendered invaluable service of 65 years in the field of education. At present, Dr. Mrs. D.P. Deshpande ma'am, Secretary, plays a crucial role in the overall progression of the Gokhale Education Society. The college has been founded in 2012 and offers MPharm in pharmaceutics and quality assurance with an intake of 15 in each. The BPharm program is permanently affiliated to Savitribai Phule Pune University, Pune, and has an intake of 100 seats. The DPharm program is affiliated to MSBT Mumbai and has an intake of 60 seats. Our college was accredited by NAC for 5 years. The college has state-of-the-art infrastructure and a lush green surrounding which provides favorable milieu for students to grow not only as good pharmacists but also world-class citizens. College has experienced and well-qualified staff with academic excellence. College maintains a tradition of good academic results and organizes various activities including conferences, seminar, student faculty development programs, workshops for overall development of students and faculty to bridge the gap of academia and industry. Coming back to today's webinar, today, the webinar titled Innovative Horizons in Pharmaceutical Sciences is brought to you by the Department of Pharmaceutical Chemistry under the aegis of Internal Quality Assurance Cell, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research. Today, we have three sessions with three eminent speakers. The session one is by an academic expert, Dr. Anindita Behere, professor in pharmaceutical chemistry, Amity University, Kolkata, who will be speaking on role of metal nanoparticles in brain targeting drug delivery. Session 2 is by Dr. Muhammad Usman Siddiqui, who is also an associate professor at SVKM's Institute of Pharmacy, Dhule. He will be speaking on understanding computer-aided drug design from basics to practical tools. And session 3 is by alumni Mr. Abhishek Dusane, who is a senior research assistant in Sai Life Sciences Limited. He will be speaking on drug discovery and development, current trends in the pharmaceutical industry. I take a pause over here to ponder upon what have been done in the last 2 years. I like to mention here this is our third consecutive year that we have been doing webinars. In 2023-24, we successfully organized 7 webinars and benefited 7,000 participants. continuing this initiative. In that time, we had only two speakers. One was from academia and one or industry, and the second was from alumni. In the second year, we started with the third session, three sessions, in which one was either academic or an industrial expert. Second was alumni of a college, and the third person was alumni of the coordinator from previous college. So, keeping on this tradition this year also, for last year statistics, I would like to mention that we organized again 7 webinars and we extended our reach to more than 13,000 delegates in the whole year only through webinars. So it is a highly impactful activity and we are continuing this today. So I welcome all the delegates and the three resource person for today's session. Now, we will begin with session 1. I request Kartiki Bandari ma'am to introduce our first speaker.
A very good morning to everyone. Today, I, Ms. Kartiki Bhandari, feel privileged to introduce our first speaker of the day, Dr. Anindita Behere ma'am. Dr. Anindita ma'am is a dedicated professor in pharmaceutical chemistry with 18 years of teaching and research experience. She is specialized in drug delivery systems and analytical techniques. She has guided two PhD scholars, numerous UG and PG students. She is passionate about academic excellence, innovation in pharmaceutical sciences, and fostering the next generation of researchers and professionals. She completed PhD from Shiksha O Anusandhan University, Orissa, M.Pharm in Pharmaceutical Chemistry from Pune University, Maharashtra, and B.Pharm at Sri Jaydev College of Pharmaceutical Sciences, Orissa. She is also qualified GATE in 2004 from IIT Delhi. Her area of interest includes nanoparticle synthesis and characterization, handling sophisticated instruments like HPLC, HPTLC, GC, AAS, FTIR, UV visible spectrophotometer, and manuscript writing. Currently, she is working as a professor in pharmaceutical chemistry at Amity University, Kolkata. Previously, she worked as an associate professor at Dayananda Sagar University, Bangalore, as an associate professor at Shiksha O Anusandhan University, Orissa, and as a lecturer at Pune University. She has various publications including 39 research papers, 2 books, and 24 book chapters. She received various awards like Award for Certificate of Merit from Justice Jugal Kishore Mahanti Smruti Sansar for securing the highest marks in the B.Pharm University examination. She is also awarded with the Certificate of Merit for Best Research Paper from Dr. P.D. Sethi Annual Award 2012. She participated as session chair for the session Advanced Waste Treatment at International Conference on Resource Sustainability, Bangkok, Thailand from 5th to 8th November 2024. She is a professional member at Pharmacy Council of India, State Pharmacy Council Orissa, the Association of Pharmaceutical Teachers of India, and Orissa Chemical Society. We are fortunate to have such an eminent personality with us today. Ma'am is going to guide us on the role of metal nanoparticles in brain targeting drug delivery. I'm sure all of us are eagerly waiting for the session. So without further ado, I request Dr. Anindita Behera ma'am to take over the session. Ma'am, please.
Thank you, ma'am, for such good introduction. So, first of all, I would like to thank to Shilpa ma'am for giving me this opportunity. Am I audible properly?
Yes, ma'am.
Okay, thank you. So, today I'm going to give some insightful means insightful uh, thinking about my research topic on which I have been working since last 8 to 9 years. So I have been working with the metal nanoparticles and I have been studying their effectiveness and their therapeutic activity in neurodegenerative diseases. Mainly, I focus on the cognitive dysfunction. So currently, I'm working as a professor at Amity Institute of Pharmacy, Kolkata. So today, my delivery includes first the introduction to metal nanoparticles, then the importances of metal nanoparticles in the drug delivery, then different types of metal nanoparticles. Next, the mechanism by which the metal nanoparticles deliver the drug through a blood-brain barrier. Then the application of metal nanoparticles for the neurodegenerative diseases, uh, application of metal nanoparticles for the brain tumor or glioblastoma, then the challenges and the limitations in the metal nanoparticles for the brain targeted drug delivery, future perspectives, and then the conclusion. So, as we know that nowadays nanotechnology has been emerging so fast because its application in various fields, not only in biomedical science, it has also application in magnetism, optical sciences, and so many different fields of science. So, generally, nanomaterials are characterized by their dimension because they have a very characteristic unique dimension having the size range of 1 to 100 nanometers, and they have been used in the biomedical science for the diagnosis, treatment, and for the prevention of many more diseases which cannot be treated by the conventional treatment techniques. So the main applications of the nanotechnology in the medicine is the targeted drug delivery, then advanced diagnostics, regenerative medicines, in cancer therapy, and the most recent is the nanorobotics. Then when we talk of the metal nanoparticles, so these metal nanoparticles mainly it comprises of the pure metal nanoparticles, but some of the nanoparticles are there which can exist as in their oxide form, and also they can be prepared for like metal organic frameworks and metal nanocomposites. So depending on their composition, they can be broadly classified as monometallic or bimetallic. So, monometallic nanoparticles have the vast application as the form of pure metal nanoparticles. Again, the pure metal nanoparticles are categorized depending upon the metal ion which we use and uh, which category they are. Like noble metal nanoparticles, like gold nanoparticles, silver nanoparticles, and platinum nanoparticles, and magnetic nanoparticle. Generally, the iron nanoparticle, copper, cobalt nanoparticles. They are generally synthesized or they show their therapeutic activity in their oxide form. Then other metal oxide nanoparticles are there like zinc oxide, titanium dioxide. So these are also other metal oxide nanoparticles which have very much good therapeutic activity, and also they are used for so many environmental remediation techniques. Then metal organic frameworks, they are composed of different metal ions with the clusters attached with each other with some linkers. And finally, the metal nanocomposites. Metal nanocomposites mainly they are made up of metals and combination of metals, non-metals, and they have the different types of characteristics like they can be alloys, they can be core cell type. So these are the vast classification of the metal nanoparticles. So as I told you that the metal nanoparticles they have very good application due to the enhanced drug stability and circulation. So generally we know that there are different kind of nanotools like polymeric nanoparticles, solid lipid nanoparticles and metal nanoparticles. All these are there, but metal nanoparticles they possess some specific unique characteristic due to which their application is much more than the other types of nanoparticles. So the gold, silver and zinc oxide, iron oxide, copper oxide, cobalt oxide, all these metal nanoparticles, they protect the drugs from the degradation when they are applied or administered by the systemic route. So they increase their half-life in the bloodstream or the plasma. So they increase their stability and the circulation time in the body. And they enhance their therapeutic activity and the bioavailability. In case of cancer or chemotherapeutic treatment, the chemotherapeutic agents they require very much targeted delivery to the cancer sites. So in those cases, two different strategies are used for the administration of anti-cancer drugs. First one is the passive targeting by which they apply the mechanism of enhanced formation and retention where they enter into the cancer cells and they show their anti-cancer activity. But the drawback of the passive targeting is that they show so much of off-target toxicity. That means they kill the cancer cells along with the normal cells also. So, much more specific chemotherapeutic drug targeting can be achieved with the help of nanotechnology by active targeting. In case of active targeting, the anti-cancer drugs, they are attached with some ligands which can specifically bind with some of the overexpressed receptors present on the cancer cells so that selectively those drug loads can only attack the cancer cells, not the normal cells. So in case of active targeting, the risk of the normal cell damage is very much reduced. Next is controlled and stimulus responsive release. So this stimulus responsive release is also a type of active targeting where the drug loadings are designed in such a way that they will release the drug only when they come in contact with some stimuli in the cancer or the tumor microenvironment. So in that case, the stimuli may be a change in the pH, change in the temperature, or the presence of some enzymes. So the benefit of this controlled and stimulus responsive uh, doses form that is that they deliver the drug in a site-specific drug release manner and they minimize the side effects and improve the therapeutic efficacy. Then multifunctionality. So when we need the multifunction with a single nano formulation, in that case we can combine multiple metals or non-metals and we can also combine with other therapeutic agents for that can be used for diagnostics, that can be used for therapy, or sometimes the diagnostics are also combined with the therapies that are known as theranostics. That is the combination of the therapy with the diagnostics. So these theranostics or these nanoformulations which are used in the theranostics, they have the benefit that along with the diagnosing the diseased area, they can deliver the drug load in that particular site with a single go. Then green and the scalable synthesis. These metal nanoparticles, they have a very good property that you can synthesize them by green synthesis method, in which we use different phytoconstituents from the different phytochemicals or some natural sources like algae, fungi, or some the plant extracts, where the phytoconstituents or the microbial constituents, they can reduce the metal ion precursors, and they can easily form the metal nanoparticles without use of any chemical reducing agent or stabilizing agent. Because when we synthesize the metal nanoparticles, we require a reducing agent to bring down the oxidation state of the metal precursor to zero oxidation state that can exist as nanoparticle form. And once the nanoparticles are formed, we need to stabilize it, because as the dimension is very less, so they have the tendency to accumulate or aggregate. Then the nano dimension may increase to micro dimension. So to prevent this agglomeration of the nanoparticles, we require a stabilizing agent. So mostly the chemicals are used as this reducing agent or stabilizing agent. But when we are using the phytoconstituent for synthesis of these metal nanoparticles, the different phytoconstituents present in the plant extracts or the microbial extracts, they itself act as reducing agent and the stabilizing agent. So those metal nanoparticles are eco-friendly, they are less hazardous, and they are more bioavailable as compared to the other methods of synthesized nanoparticles. So today I am focusing on how the metal nanoparticles can be used for brain targeted drug delivery. So we know that when we think of designing a drug delivery system for which can target the brain cells, then there may be three different types of delivery system. One is the local delivery, in which we can directly inject the formulation to the meninges. Second one is the intranasal delivery, in that the drugs can be transported to the brain via the olfactory bulb located at the top of the nasal cavity. And the third one is the systemic delivery, in which we inject the drug into the bloodstream, and the drug has to cross the blood-brain barrier, and it has to reach to the brain cells. So today my focus of the discussion is this systematic delivery, because if I will cover all these three routes of delivery, then it will take a very long session. That's why I have taken the systemic delivery approach, by which we can design the different brain targeting drug delivery systems. So before going in detail, we should understand why the permeability through the blood-brain barrier is a biggest challenge when we are designing the brain targeting drug delivery. So for that, we need to understand the composition of the blood-brain barrier. So the blood-brain barrier, that is, in simple words, you can tell that is a separating barrier between the brain cells and the blood capillaries or the blood supply. So mainly the blood-brain barrier, it is a combination of three different membranes. One is the endothelium of the brain microvessels. Then another is the epithelium of the choroid plexus and the epithelium of the arachnoid matter. And the neurovascular unit of the blood-brain barrier, it is made up of endothelial cells, microglial cells, pericytes, and astrocytes. So these endothelial cells, you can see in this diagram I have shown here. Is my cursor visible here? Is my cursor visible, ma'am? Okay, so you can see this is the capillary in the middle part. The capillary lumen is there. Around it, the endothelial cells are there. These endothelial cells, they are forming the tight junctions of the blood-brain barrier. So one longitudinal section is shown in the lower part of the figure where you can see the this purple color cells. They are the astrocyte and foot. Below that, the tight junction of the blood-brain barrier is there, which is separating the blood supply from the brain cells. So, to administer the drug through the blood-brain barrier, the drug is transported to the brain cells from the blood supply or from the systemic circulation by different transport mechanisms. So, mainly three mechanisms are there. One is carrier mediated transport, another one is receptor mediated transcytosis, another is adsorptive mediated transcytosis. So, this carrier mediated transport, it carries different kinds of small nutrient molecules like ascorbic acid, glucose, glutathione, and tyrosine, mainly the different amino acids that are required for the brain cells. So, but when we are delivering the drug, so the drugs are generally macromolecules. That's why they cannot be transported through the carrier mediated transport. So the drug, when that is delivered to the brain cells, they are transported to the brain cells through the receptor mediated transcytosis or adsorptive mediated transcytosis. This is one detailed uh, diagram that represents the different types of the transport mechanisms through which the drug is passing from the systemic circulation into the brain cells. First one is the paracellular pathway. This is used through this transport mechanism. Very small molecule, drug molecule having their molecular weight less than 400 kilodalton, they can pass through the tight junctions of the blood-brain barrier. Second one is the passive transmembrane diffusion. That passing of the drug, it is a concentration gradient dependent. So in the systemic circulation where the drug concentration is high and the brain cells where the drug has to move, their concentration is less. That's why there is a concentration gradient between the blood and the brain cells. So the drug is moving from higher concentration side, that is from the blood, to the lower concentration side, that is to the brain cells. So that is the mechanism by which the passive transmembrane diffusion is taking place. Then carrier mediated transport, as I told, these are mainly useful for the transport of the small molecules or amino acids. So here there is a carrier which will take the small molecules and that will release the molecule into the this epithelial cells. Again, on the brain side, the again another carrier sites are there which will drag those drug or moieties. Then again they will release through this carrier. Fourth one is the receptor mediated transcytosis, and the fifth one is the adsorptive mediated transcytosis. These two, this receptor mediated transcytosis and the adsorptive mediated transcytosis, these are the two major pathways through which the metal nanoparticles pass through the blood-brain barrier. So, in the receptor mediated transcytosis, there are receptors present on the luminal luminal side of the blood-brain barrier, which will selectively attract or which will selectively attach with the drug molecules. Then, through a endocytosis process, the receptors, they release the drug into the brain cells. But the drawback of the receptor mediated transcytosis is that when we uh, continuously administer the drugs, sometimes the receptors get saturated and they are no more functional in taking the drug from the blood circulation and to deliver in the brain cells. Last one is the absorptive mediated transcytosis. It's a very effective mode of transport because it works on the surface charge present on the metal nanoparticles, and the plasma membrane, the epithelial cell membrane, they interact with those charged surfaces of the nanoparticles and they can easily pass them through the epithelial cells into the brain cells. So when we talk of the application of the metal nanoparticles for the brain targeting drug delivery, so mainly three aspects are there. First one, we can use the metal nanoparticles for imaging. Second one, we can use the metal nanoparticles for therapy. And third one, we can combine the imaging and the therapy and we can approach for a theranostic activity of the metal nanoparticles. So when we use them for the imaging technique, so imaging technique, it has a very wide application in the brain diseases because the compactness of the brain cells, it makes very difficult to identify the diseases through normal scanning techniques. That's why when we expose the brain cells to these highly effective metal nanoparticles in the imaging technique, then it is very much effective because the metal nanoparticles, due to their dimension in the nano range and their shape and size can be easily tunable, which will modify their physicochemical property, and they can be helpful for the different imaging techniques. So generally MRI is used for the imaging of the brain or the CNS system. So when we use the metal nanoparticles, then their contrast agent which is used or which is using the nanoparticles, mainly gold nanoparticles, iron oxide nanoparticles, and gadolinium. They are significantly used as the contrast imaging reagents for the MRI, then CT, and the PET scanning, and they enable the very clear visualization of the CNS structures. Then the next benefit of using this metal nanoparticles in CNS imaging is that they can easily penetrate through the blood-brain barrier because the metal nanoparticles sometimes they are functionalized or they are surface coated with some other polymers or some other agent which can increase their permeability through the blood-brain barrier, or itself, if we talk of the gold nanoparticle, the gold nanoparticles are very easily permeable through the blood-brain barrier. So, these metal nanoparticles, they have the advantage that they can enter through the blood-brain barrier by non-invasive techniques. That's why nowadays these nanoparticles are in much more demand for the non-invasive imaging of the brain and to detect the different types of abnormalities or the protein aggregations or the tumor cells, or to identify the plaques in case of Alzheimer's disease. Then they can be combined with multimodal imaging capabilities, like you can combine MRI with CT or you can combine MRI with PET scan. So what happens when we combine all this uh, metal nanoparticles, then they are capable of identifying or diagnosing the brain cells or the CNS abnormalities with higher accuracy, and they can give comprehensive CNS imaging. And another benefit is that targeted imaging. So when we have the brain tumor or the glioblastoma, so instead of exposing all the brain cells, we can use some radio-labeled metal nanoparticles as contrast agent, and we can identify the location where the glioblastoma or the tumor cells are growing. So they are used for the disease markers in the brain, and they improve the localization and specificity of the imaging conditions in glioblastoma and Parkinson's disease. Next advantage is, by this imaging techniques, these advanced imaging techniques using the metal nanoparticles, we can diagnose the real time or the early diagnosis of so many CNS abnormalities. So nowadays, those which are diagnosed with pre-Alzheimer disease symptoms, nowadays they are subjected to this real time and the early diagnosis techniques. For that, before the disease will increase in its intensity, we can identify and we can start the treatment of the patient. Then the next application is in the therapy. So the different mechanisms by which the metal nanoparticles, they are in much more use for the brain targeting therapy are due to the enhanced blood-brain barrier penetration or permeability due to their tunable size and dimension. They have very good antioxidant and neuroprotective properties. They have very targeted drug delivery systems, and they can be used for the theranostic applications by combination with the diagnosis with the therapy. So here are some of the mechanisms here I have discussed. It has enhanced BBB permeability, and these metal nanoparticles, they are having very good intrinsic antioxidant activity. They can neutralize the reactive oxygen species and reduce the oxidative stress related the metabolic pathways in the CNS. Then they can deliver the drug through a targeted delivery system if we combine them with some ligands, and they can be used to uh, target the amyloid beta proteins and the alpha synuclein in case of Alzheimer disease, and they can be used for site specific delivery of the drugs or the gene therapy components. And due to this targeted delivery, they have very less off-target side effect. And these metal nanoparticles have a very nice or good anti-inflammatory activity. Due to that, the different pro-inflammatory cytokine levels are decreased, and they can be very much helpful in reducing the neuroinflammation, neurodegeneration, in case of diseases like ALS and Huntington's disease. And in theranostic application, we can, in a single shot, we can identify and we can start the therapy with the metal nanoparticles in the CNS region. So here are some schematic representations of the mechanisms by which the metal nanoparticles or the gold nanoparticles, they are improving the symptoms of Alzheimer's disease and the uh, Parkinson disease. Okay, so in case of Alzheimer disease, they prevent the anti means aggregation. Then they also have the neuroprotective activity. And in case of PD, they have uh, this anti-inflammatory activity. They promote the neurogenesis. New neurons will be generated, and they reduce the oxidative stress, and they inhibit the alpha synucleation aggregation. So currently, I am engaged with the study of the cognitive impairment or the AD-like symptoms in diabetes patients, because in chronic diabetes, after uh, some years of the diabetes, the brain cells, they have the also they have the insulin resistance, due to which there are cognitive decline. And for that, in that, I have studied the application of different nanoparticles, and I have published papers in different peer-reviewed journals. So I have synthesized so many monometallic nanoparticles, bimetallic nanoparticles, and these are some recent publications I have done in JDDSC, molecular neurobiology, scientific reports, experimental neurology, and this is one of the recent paper I have published this year, where I have compared the two active ingredients. One is the natural molecule a flavonoid hesperidine, another one is a synthetic drug vildagliptin. So for both the drugs, I have synthesized monometallic nanoparticles like gold and selenium nanoparticles, and also I have synthesized their bimetallic selenium on gold. And I have studied their activity and compared the effectiveness in cognitive impairment in a STZ-induced diabetes rat model. So in this study, we found out that though the bioflavonoid has a very nice antioxidant activity, but the overall activity, if we will consider for the cognitive impairment, then the vildagliptin conjugated nanoparticles, the bimetallic nanoparticles, they exhibited very significant improved cognition in the rat model. This is published in the Journal of Neuroimmunopharmacology this year. Next, we will discuss the application of metal nanoparticles in the brain tumor or glioblastoma. So, as I told, then in cancer treatment, we can go either with passive targeting strategy or active targeting strategy. But due to the more site-specific delivery of the drug to the cancer cells, mostly the active targeting strategies are adaptable. So, for that, we combine the chemotherapeutic agents or the anti-cancer drugs with some antibodies or some other ligands which are very much receptor specific, and when the anti-cancer agents are ligated with the ligands, they specifically bind with those receptors present in the on only cancer cells, not the normal cells. That's why they have very good therapeutic efficacy. They prevent the multi-drug resistance, and they also have very less systemic uh, toxicity. Then the next application of the metal nanoparticle is the photothermal and the photodynamic therapy. So many cancer treatments, they require the photothermal therapy or the photodynamic therapy, in which with the help of the heat they can also kill the cancer cells. And in the photodynamic therapy, this specific metal nanoparticles, they enter into the cancer cells and they increase the reactive oxygen species so that the cancer cells will die. So, this specific photothermal and photodynamic therapies are very much effective with the multi-component or nanocomposites, because sometimes the monometallic nanoparticles, they are less effective as compared to the multi-component or bimetallic nanoparticles, because the bimetallic nanoparticles or nanocomposites, they show some additive or synergistic effect to kill the cancer cells. And the last one is your theranostic. with the MRI techniques. Here, mainly the gadolinium-based nanoparticles, they are used as the MRI contrast, and they allow the real-time imaging. And real-time imaging means when we use these techniques, we can visualize what is the exact position or what is the exact intensity of the tumor in the brain. And immediately we can administer the required drug in form of nanoparticle, because due to their size they can easily pass through the blood-brain barrier, and it's sometimes these are the non-invasive techniques. That's why the patient is not treated with any difficult surgeries. That's why these are very good for diagnosis and the treatment simultaneously. These are some of the literatures I have got. So for your reference, here, this is the passive targeting by which the enhanced permeation and retention driven delivery of 5 nanometer magnetic iron oxide nanoparticles in an orthotopic murine model of glioblastoma. So here you can see this is the tumor cells, and beside that the normal brain cells are there. So here the metal nanoparticle that is injected into this murine model of the glioblastoma, and where the nanoparticles are going and they are entering into this tumor zone, and you can visualize with the confocal microscopy they have identified the nanoparticles, they are entering into the tumor cells, not the normal cells. But sometimes the long treatment with the passive targeting strategy that shows the systemic toxicity or of target uh, neuronal death. That's why the next diagram which I have given here, that is for the active targeting, where these are the different receptors which can be targeted, and by targeting these overexpressed receptors, we can design the nanoformulation or the nanoparticles with the metals in such a way that the drug can easily go and bind with this receptor, and they can only enter to the cancer cell. They will not affect the normal brain cells. Next, the challenges and the limitations. So the big challenge is the designing the drug delivery which have the very good BBB permeability or penetration. So though lakhs of scientists and researchers are involved in this research, but till now nobody has cracked this problem or the limitation, that how easily they can design the drug delivery system which can easily go through the BBB. Again, the metal nanoparticles, they have the limitation of the toxicity, neurotoxicity and the biocompatibility, because when we talk of the size, the tunable size and the physicochemical properties of the nanoparticle which is effective in therapy, sometimes that size becomes so small that it can enter into the brain cells in a such big concentration, so that instead of neuroprotective effect, it may cause the accumulation of the metals, and that may cause neurotoxicity. So, as the gold nanoparticles have very wide application for the brain targeting drug delivery, but there are certain uh, means literature citations where they have cited that the size of gold nanoparticles below 5 nanometer, they can produce neurotoxicity. Again, the size above 50 nanometer, they cannot also penetrate through the blood-brain barrier. So the size above 5 to 15 nanometer are the most suitable size of the gold nanoparticles which can show the effective therapeutic activity in the CNS cells. Next challenge is the clearance and the accumulation. So when we administer these metal nanoparticles with the systemic circulation, so before reaching the blood-brain barrier, they may be interacting with the macrophages. and their availability is decreasing to the blood-brain barrier site. Another limitation is that they start accumulating and they become bigger in size. So, when they reach the blood brain barrier, they cannot pass through the BBB barrier. Then, the another is that their toxicity due to accumulation in the liver, because the liver is the main metabolizing organ in the body. So when we administer these metal nanoparticles, the metal nanoparticles, they accumulate in this liver and spleen and they may cause toxicity to that. Lastly, the major uh, difficulty in case of metal nanoparticles for the brain targeting drug delivery is that their scale up and reproducibility and the cost, because there is a very big challenge regarding the reproducibility in the batch to batch manufacturing of these metal nanoparticles. Next is, what are the future perspectives that we can look forward with the metal nanoparticles? First one is the precision engineering for the BBB navigation. We can design the stimulus responsive metal nanoparticles which will categorically deliver the drug load when they come in contact with some stimuli. And we can also design ligand conjugated metal nanoparticles and which will deliver the required quantity of the drug to the patient. In precision drug delivery, when we talk of regarding with respect to brain, so each and every patient who are suffering from neurodegenerative diseases or brain tumor, they require different doses of drug for their effective therapeutic activity. So we can design those required doses nanoformulation with the help of the AI techniques, and we can also tailor the nanoparticles, metal nanoparticles with other means surface active reagents like different polymers we can use, and this study is in vast application nowadays. And we can target for different theranostic applications which will enhance the diagnosis and the targeted drug delivery to the brain. Then the last one is the designing some biodegradable and safer metal nanoparticles with the green synthesis. So the green synthesized metal nanoparticles, they are biodegradable, they are eco-friendly, and they are less costly and less hazardous. So lastly, I would conclude that the metal nanoparticles, they are very efficient in brain targeting drug delivery by overcoming the limitations of the blood-brain barrier permeability. Then multifunctionality due to their unique physicochemical properties and the different types of their sizes and their shapes, we can use them as for imaging, for therapeutic activity, and also as theranostic applications. They have very increased therapeutic efficacy due to their enhanced bioavailability, enhanced cellular uptake, and site-specific release of the neurotherapeutics or chemotherapeutic reagents. Then, though there are sort safety and the toxicity concerns, we still we need to work a lot so that we can overcome the challenges for these metal nanoparticles when we want to deliver it to the brain cells. And future ready technology with the AI, we can design the biodegradable and personalized nanomedicines which can revolutionize the brain targeted drug delivery. These are some of the references from which I have prepared my presentation, and thank you a lot for your patient hearing, And if any questions are there, I welcome them all.
Thank you so much, ma'am. It was indeed an informative session. Now the session is open for discussion. If participants have any questions, you can unmute yourself and ask, or you can put it in the chat box. Ma'am, we have one question.
Yes.
How can we distinguish between neuroprotection and anti-cancer effect of synthesized compound in SHSY55 cell line?
Yes. So the cell line, thank you, Mr. Hemant, for the question. So it's a very interesting question. In case of this cell line study, this is a in vitro study by which we generally we assess the anti-cancer activity of different kind of molecules. So we use this cell line study for the assessment of the efficacy of the different nanoparticle or the nano formulations. So neuroprotection activity you can assess through the antioxidant study, and you can also check with the levels of the pro-inflammatory cytokines like your TNF alpha, IL-6, IL-1 beta. Those levels you can check, and nitrite level, then cholinesterase level. Those are the biochemical estimations you can do for the neuroprotection. And for anti-cancer study, you can go with the MTT assay of this cell line study, and you can also go for the different other techniques which can assess the anti-cancer activity, like the measurement of different levels of the biochemicals, like some cancer stimulating hormones are there. So those levels you can check for the anti-cancer efficacy of the nanoparticles. I think I am able to answer the question, or is there anything left?
Yes, ma'am. Thank you so much for the answer. Okay, there are no further questions, so let's move forward.
Okay, thank you, ma'am. Shall I leave?
No, ma'am, just wait a minute.
Okay, okay, thank
I would like to thank you, ma'am, for taking out time from your busy schedule and guiding us through your insightful session. Ma'am covered all the aspects of metal nanoparticles, their types, mechanisms, challenges, future perspectives, and applications with various examples in detail. On behalf of Gokhale Education Society, Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, I thank you and request you to accept the certificate of appreciation as a token of love. Once again, thank you so much, ma'am.
you. It's my
Now we
honor.
will,
Thanks a lot.
yes, ma'am, thank you. Now we will take a break and come back at